Hello viewers, today we will discuss about the another very important disorder which is related to the central nervous system, and the name as you can see on the board is Myasthenia gravis. तो आज हम एक और सेंट्रल नर्वस सिस्टम डिसऑर्डर की डिजीज की बात करेंगे जिसका नाम Myasthenia gravis है। तो इससे पहले कि हम इसकी डिटेल्स में इसकी इन डेप्थ एनालिसिस हम स्टडी करें, इससे पहले हमें ये देखना होगा कि एक्चुअली मस्कुलर मस्थीनिया ग्रेविस किस की पैथोफिजियोलॉजी क्या है। तो पैथोफिजियोलॉजी, सो एज वी ऑल नो दैट मस्थीनिया ग्रेविस इज अ डिजीज ऑफ न्यूरो मस्कुलर जंक्शन disease. So as we all know that that particular disease known as Myasthenia gravis is a disorder of neuromuscular junction, and it is a autoimmune disorder, right? It is a autoimmune disease. तो क्योंकि हमें ये पता है कि ये एक न्यूरोमस्कुलर जंक्शन डिसऑर्डर है और the primary cause of this particular disease is it develops some kind of antibody, autoantibody against the some kind of neuromuscular junction. So before we are going into the detail of that specific Myasthenia gravis disorder, first we should know that what is the physiology of neuromuscular junction, right? So as you can see that this is nothing but a spinal cord from where the lower motor neuron is started, right? So this is the central nervous system part, spinal cord, from where the cell this any nerve is coming, that is motor nerve. This is a motor nerve coming from the spinal cord, and it reaches up to the sarcolemma of the muscles. This is a sarcolemma. So this is nothing but a neuron which is attached to the muscle, right? So you can see this is the so what we call this is presynaptic. This is presynaptic formation of that neuromuscular junction, and this is the sarcolemma, right? This is this whole structure from here to here, this known as sarcolemma, right? And in between there is a space in between which is known as cleft, right? Which is known as cleft, synaptic cleft. Okay, this is space. It's neuron and this sarcolemma. What we call this space, it is synaptic cleft. Right? So this is all about the if we divide the neuromuscular junction into three parts. So we have presynaptic part, then we have distance, or we can say space between the presynaptic and postsynaptic, that is sarcolemma. Now what happens whenever any action has to be occur, that the central nervous system send the signal through the nerve, that is a motor nerve, right? This is a motor nerve, right? So how can we send any signal? It has to be coming from the central nervous system in the form of action potential, right? It is coming into the action potential form. But when it reaches to that presynaptic neuron, so we have certain kinds of channels, right, which open when this action potential coming through the central nervous system. There is an action potential generated that we have to move. For example, if I have to pick a pen, so my central nervous system cortex is telling that I have to act in this manner to get to take a pen. So the signal is coming from my cortex to the spinal cord, and from the spinal cord there is a motor nerve which get the action potential up to this presynaptic neuron. Now when it reaches up to this, so you can see this pinkish channel. So this is nothing but a sodium gated channel. This is nothing but a sodium channel. So as the name indicated, when the action potential is reaches here, it has to be closed. It is already closed, but when the action potential is coming here, it opens. And as we all know that there are so much sodium extracellular, where is the concentration of sodium ion is more? Yes, it is absolutely, it is outside the cell. So when the action potential reaches here, the sodium channel is open and lots of sodium is going inside, right? It is going inside because the sodium channel is open. Now next to this, you can also see another orange channel, which is nothing but calcium. This channel is nothing but calcium voltage gated channel, right? So as the action potential is coming from the spinal cord, reaches up to this level, this is nothing but a sodium channel, right? This is nothing but a sodium channel. And when reaction potential is coming here, the sodium channel is open and lots of sodium coming inside. So when the sodium is coming inside, what happens to that neuron? Obviously the resting membrane potential will be more, right? And there is a calcium voltage channel is inside, which is very sensitive to action potential. And because the action potential is, the resting membrane potential is increases, these channels now. So we are discussing about the action potential which is coming from the spinal cord, reaches up to the sodium gated channel, which is open, and then there is lots of cation inside the neuron. And then because we have calcium voltage gated channel, because of the action potential, or we can say resting membrane potential is increases. So with that, these calcium voltage gated channel are now open, and when it is open, so we can easily understand that the lots of calcium ion is coming inside. But the real point is that why we need these calcium inside, क्योंकि अगर हम देखें तो ऑलरेडी वी हैव कैटाइन इन द फॉर्म ऑफ सोडियम, लेकिन अब हमें कैल्शियम की क्यों जरूरत है? सो फॉर दैट यू कैन सी इन दिस circle, it is nothing but a vacuole present in the terminal part of the neuron, which is having acetylcholine in the form of a green dot. You can see in this diagram easily. These are nothing but the acetylcholine, which is a neurotransmitter. Okay, so you also can see that there is some kind of projection which is coming from the vacuole. एक आपको एक vacuole यहाँ पर दिख रहा होगा, जो कि एक प्रोटीन होता है जो वैक्यूल के जो मेम्ब्रेन होती है उससे अटैच होता है. इसका ये फंक्शन होता है कि इट हैज़ टू बी अटैच टू द न्यूरोलेमा ऑफ अ न्यूरॉन. उसको समझने के लिए अब हम अगर इसको थोड़ा मैग्नीफाइंग कर देते हैं, सो इट इज़ नथिंग बट अगेन द वैक्यूल विच इज़ कंसिस्टिंग ऑफ lots of, these are nothing but acetylcholine, we can say, right? These are bubbles of acetylcholine or neurotransmitter which has to be released at the terminal end, or we can say the synaptic cleft from जहाँ पर we have so many, ये आपको structure दिख रहे होंगे, these are nothing but acetylcholine receptors. Acetylcholine receptors. तो यहाँ पर इन रिसेप्टर से अटैच होने के लिए we need to release the acetylcholine. ठीक है. So now we magnifying this part here. So you can see this is a protein which is attached to the membrane of vacuole, and there is another we can say lock and key type of projection which is present at the neurolemma like this. Okay, so it has to be attached, and then exocytosis occur and acetylcholine is released into the synaptic cleft. So again you come to the point of calcium ion has to be come inside because of opening of calcium voltage gated channel. But जो पॉइंट है कि ये हमें ज़रूरत क्यों होती है, सो इसको समझने के लिए जब हम इधर आते हैं तो ये जो इसका लॉक एंड की प्रोजेक्शन अनदर प्रोटीन है जिसके साथ इसको कनेक्ट होना है, एंड देन द एक्सोसाइटोसिस प्रोसेस कैन बी डन. सो इनिशियली व्हाट हैपेंस, these projection which is present on these lock and key part, it is like this, right? For opening of these projection, or we can say to connect these two clumps, it has to be removed. These protein, these projection has to be removed, which is only when we have calcium binding on these sides. ये तभी हो सकता है जब ये कैल्शियम इस साइड से बंध जाए, राइट, या बाइंड हो जाए. सो एज सुन एज द कैल्शियम आयन इज कमिंग इनसाइड, इट बाइंड्स टू दैट पर्टिकुलर साइड, एंड नाउ आफ्टर दिस binding of calcium, it has to be open, and now it is free to bind with the vacuoles. And you can see here, after this it has to be attached to this, and finally the exocytosis process occur, and by the exocytosis the acetylcholine is released, right? I think up to that level you understand. Now what happens after this? Now we have acetylcholine in the synaptic cleft. Now these acetylcholine, where it has to be attached, it obviously it has to be attached to the acetylcholine receptor. To understand this, we can make the diagram in a magnifying manner. Okay, we can make it here. For example, this is the acetylcholine receptors, and for example this is the acetylcholine is attached here. So when these acetylcholine attached to the receptor, so what is the function? The function is that it has to be open. So when the acetylcholine is here, so the gate is open, and lots of cation is going inside, right, through these channel. Okay, okay, so this is the process of this is the function of acetylcholine, that it has to be attached to the receptor, acetylcholine receptor. And because when these receptors open, there are lots of cation is going inside. इससे क्या होता है, बहुत सारा cation हमारा inside the sarcolemma has to be entered, and we can understand what is the resting membrane potential. Resting membrane potential of sarcolemma is minus 90 millivolt, right? It is minus 90 millivolt. एंड जब यहाँ पर कैटाइन एंटर होने लगेगा तो उसके नतीजे में क्या होगा, दिस रेस्टिंग मेम्ब्रेन पोटेंशियल कैन बी 60 or 50 or like this, right? So initially what happens, there is a local action potential is generated, and finally when lots of cation is going inside, so this action potential has to be traveled throughout the muscles, and the muscle is contract. This is how this action potential from the cortex to spinal cord up to the muscle has to be reached. This is the full physiological process. Apart from that, because we are discussing the Myasthenia gravis, another very important, these receptors. So we are talking about the receptors. So there is one important protein. You can see this guy is sitting on this sarcolemma, one of the crest. This guy is known as musk, right? Or we can say musk protein. Now अब हमें ये समझना होगा कि why it is very important, why is sitting on this these crest. So musk protein function is that it regulate. What is the function of this protein? It regulate the concentration, or we can say density of these receptors. What are these receptors? Acetylcholine receptors. So you just remember this musk protein, because when we discussing about the pathology of Myasthenia gravis, so you have to remember this, because it is a very important function. As we can we can just understand that it has these protein main function is to regulate the number of receptors, right? जैसे हम समझते हैं, जैसे हमें मालूम है कि जब भी कोई भी फंक्शन हमारे लिविंग सेल में होता है तो उसमें वियर एंड टियर होता है, राइट? सो दीज रिसेप्टर्स आर आल्सो गेटिंग ओल्डर एंड दे हैज टू बी रिप्लेस्ड, राइट? सो हु इज रिस्पॉन्सिबल? दिस इज मस्क प्रोटीन इज रिस्पॉन्सिबल. व्हाट इज द फुल फॉर्म? इट इज नथिंग बट मसल स्पेसिफिक काइनिन प्रोटीन, राइट? Okay, so this is all about the physiology of neuromuscular junction and how the action potential come. Okay, one point is remaining. You can have you see this bird is sitting on this crest? Oh, this bird, have you seen this? And that bird is chewing these green dots. And what is the green dots? Green dots are nothing but acetylcholine. ये जो bird है, it is nothing but a acetylcholinesterase, right? What is the name of this bird? Acetylcholinesterase. होता क्या है, जैसे ये एसिटाइलकोलिन रिलीज होता है और ये अपना फंक्शन यहाँ पर मुकम्मल करता है, ये अपने रिसेप्टर पर अटैच होता है, तो इट शुड नॉट बी देयर फॉर लॉन्ग टाइम, और हम कह सकते हैं कि ये हमेशा यहाँ नहीं रह सकता. उसके लिए यहाँ पर एक एंजाइम होता है जिसका नाम होता है एसिटाइलकोलिनेस्ट्रेज. व्हेन द फंक्शन ऑफ दिस एसिटाइलकोलिन इज कंप्लीटेड, इट हैज टू बी रिमूव विद द हेल्प ऑफ दिस enzyme which is known as cholinesterase, acetylcholinesterase, right? So you also remember this enzyme, right? It is very important, as when we discussing about the therapeutic or treatment part of the Myasthenia gravis, this enzyme is very important, and we can say it play a very important role. So I hope that you understand physiology of neuromuscular junction. So the next part we will discuss the pathophysiology of Myasthenia gravis. This is very important because now you can easily understand what actually happens if you know the normal function, right? Okay, thank you for watching. If you like this video and if it is informative, you just subscribe our channel and like the video. Thank you very much for watching.